Ethan, thank you so much for finding the time today and coming on to talk to us about your work. It's great to have you.
Thank you. Good to be here.
So your recent research focused on the genetic modification of insects, specifically the fruit fly. You've been working on developing a technique that's called gene drive, which allows you to spread a specific genetic change through an entire population of insects. So, can you first tell us why it is important for us to be able to modify the genes of such insects?
Well, the reason, Alistair, is that you have certain problems that we're facing with insects that are very, very challenging with regard to the use of traditional tools. So, as you're probably aware, the most commonly used way of controlling insects is with insecticides. And so if you look at a disease like malaria, because we're also very interested in potential use of this technology to reduce the incidence of malaria, in the last 10, 20 years, there's been enormous success in reducing the deaths by about 50%. And those gains have been made largely by using insecticide impregnated bed nets. So in the fabric, you have the insecticide chemically linked. And then they also have a long-lasting spray that they put on indoor walls. And together that has reduced the number of insects sufficiently so that, the number of malarial deaths, for example, has gone down by a factor of about 2. But over the last 3 or 4 years, and particularly under the conditions of the pandemic, that it's no longer going down. And then the question is it just level, or is it maybe going back up? And the reason that is happening, and it also pertains, the same kind of logic pertains to the control of crop pests, which cause, food insecurity. That's another problem that also happens with insects, is that they've developed resistance to insecticides. And if you look, for example, in Africa, there's a particular mutation in mosquitoes, but also in other insects as well, that keeps repeatedly getting changed from one amino acid residue in one particular part of this protein that is the target of the insecticide. It's an ion channel that's necessary for the function of the nervous system, and it's the ion channel to which insecticides like the old one, DDT, or the more recent ones, pyrethroids, bind to, and then they cause a defect in the functioning of that ion channel that causes the animal to die. And what's happened is they simply have acquired mutations in that target site that render it impossible for the insecticide to bind anymore. It no longer physically binds to the ion channel, and so they're resistant. There are other resistance mechanisms on top of that too, that insects have evolved ways of degrading the insecticide more rapidly, or they thicken the cuticle, the outer covering of the insects. That's how they get poisoned by it. Basically, they land on it, and then the chemical gets into them that way. And so they just made the equivalent of their skin, their cuticle, which is like a fingernail, a little bit thicker. And so that also makes it more difficult to kill them. So by these various combined ways, it's come to the point where the bed nets are no longer really effective, and the mosquitoes then are finding the little holes in the nets, and then they're getting in. And so the idea is that we needed some alternative approach that could be used still in combination with the existing tools like bed nets, and the same thing is also true in terms of resistance with the drugs that people use against, say, malarial parasites or something. The parasites are getting resistant. So you just have to have all these tools working together, and the thought was to add another one wherein what you do is you transform genetically the insect so that it's no longer so much of a problem from a health perspective or from a crop pest perspective.
Can you talk a bit about how the genes mutate so that insects actually build up this resistance to the commonly used insecticides?
Well, again, a good example of that is this target site mutation that I mentioned, the actual place where the insecticide binds. So in Africa, in 12 independent populations of insects, the same mutation has arisen. And what we think goes on is that these changes that happen in the genome, they're happening every generation of every organism, and human beings too, from one generation to the next. There are a few changes that are made. And of course, with the principles of Darwinian selection and so forth, the idea is that most of those mutations or changes are either neutral or deleterious, and they get weeded out largely or not amplified. But occasionally a mutation gets made that's in exactly the right place, and then that one gets amplified because those organisms that carry that have a great selective advantage. And it's hard to come up with a stronger selection than an insecticide, because if you don't have the mutation, you're dead. And if you do have the mutation, you live. So that's a very, very strong form of selection. And so in this particular case, since the target site mutation turns out to be an important element of the selection process by which insects become resistant, that mutation has arisen over and over again independently. And so we think that that's a good place to start for intervening in a genetic way to say, well, if there's you could say a good version of the gene, which is the version of the gene that could be targeted with insecticides, and a bad version of the gene, which is the version of the gene that renders them resistant, could we convert the bad version to the good version? That was kind of the thinking that led us to this work.
And let's talk a bit about your work that you did in more detail. What are the specific genetic changes that your team introduced in fruit flies?
Right. Well, there I could just maybe back up a little bit and mention what a gene drive idea is, and then how we piggyback on that concept for what we call an allelic drive. So a gene drive is a genetic cassette that contains essentially molecular scissors. And those molecular scissors are programmed through this whole CRISPR revolution to be able to cut the genome exactly at the place where you where you insert the scissors into the genome. So what happens when you do that is that if say a mom carries this trait and she mates with a male that doesn't, then in the offspring that come from that union, the gene from the mother, which is inserted in the chromosome exactly at the place where it's programmed to cut, will see the father's chromosome as unmodified, and it will cut it. And then through a process by which normal DNA gets repaired, during sexual reproduction, chromosomes are cut many times and rejoined to shuffle genetic material between the mother and the father. So this is an inherent feature of sort of what they call meiotic repair, which is what happens during the process of meiosis to produce the germ cells of the organism. So they're prepared to do that. It works with the biology of the organism. And what happens is that during that repair process, the cassette just simply gets copied. So now, instead of having only 50% of their offspring inheriting the trait, which would be the normal situation, all of them do, or almost all of them do. And remarkably, that does work incredibly well, so that it really does get up into the 99% levels or so forth, that you can have that happen. So that once you have that going on in one part of the genome, then the idea is, well, you can piggyback on that with the same kind of concept and now trigger the scissors not only to cut at the point where they're copying the genetic information, but also to cut in another location in the genome. And there, the idea is what you do is you cut the bad form of the gene that you don't like, the one that would say favor parasite transmission or render the insecticide resistant in this case. But it does not cut the favored versions. So it doesn't cut, in the case of the insecticide resistance example, it doesn't cut the version of the gene that is sensitive to the insecticide. So now this genetic element, which is prepared to spread itself through a population because it copies itself every time it gets inherited and goes to all of the offspring. At the same time it does that, if it encounters the bad allele, that allele is a version of a gene, if it copies the bad version of the gene that renders the insect resistant to insecticides, then it cuts that version. And you would, of course, generate such an insect in a genetic background that is sensitive to the insecticide. So it would carry a sensitive version of the gene at the same time. And now it corrects the cutting that it's done in the bad gene by copying the information from the good gene. And so now all the progeny not only inherit the genetic cassette in this case, but they also inherit the preferred naturally occurring variant of the gene, which is in this case, the insecticide sensitive version of the gene, which is the normal version.
And how do you ensure when you do all that the genetic modification only affects the targeted insect population and not other organisms?
That's a good question. And in general, with gene drives, you've got a lot working for you, because you can decide where in the genome you want to insert it. And the genomes of different insects, even closely allied insects within the same narrow family, the genus, they will have differences, many, many differences across their genome. So you just insert it in a region that is specific to your target species. If it somehow got into another closely related species, because you can make hybrids sometimes, at least within the genus, you don't really make hybrids very often outside of the genus, but within the genus you can. But even if you did, it wouldn't be able to cut because the site wouldn't be there. So the scissors wouldn't be able to cut. They'd run into a knot on the other end. And then the beauty about the allelic drive is you have two such safeguards. You have the safeguard in terms of where you put it to begin with, and then almost always, even the target site that is being mutated, it will be different among different insects. Within the same genus, maybe they'd be the same or similar, but certainly outside of that, they'd be different. So you'd have a kind of double lock on that. So you could be pretty certain that you'd be target specific in terms of the species.
So that sounds all really exciting. And you did this work in fruit flies. The mosquitoes are a great concern for diseases like malaria. So what is the next step now for you in terms of this work, and also bringing it to broader adoption, maybe?
Those are both good questions. So with regard to mosquitoes, of course, we still have this issue of insecticide resistance being a big problem. So that whole insecticide, the whole idea of reversing insecticide resistance in mosquitoes pertains to mosquitoes just as well as to fruit flies or to other crop pests. So that's a transferable kind of technology. In the case of mosquitoes too, though, there's another exciting opportunity that uses exactly the same technology that we're working on. And in that case, the idea is that there's a mosquito gene that is required for the parasite, the malarial parasite, which enters through the blood of the person it's bitten. It gets into the gut of the mosquito, and then it crosses from the gut into the body of the mosquito, and then it makes its way to the salivary glands, where it then can get inserted into the next poor human being that gets bitten. But it has to cross that epithelial barrier between the gut and the body wall, and to do that, it must bind a protein called FREP1. It's just the name of that protein. And that protein exists in two different forms, allelic forms, the two different variants of it, one of which the parasite can bind to, and that helps it get across. And the other version, which is fully functional from the point of view of the mosquito, because the mosquito needs this protein to be healthy, but it's completely fine for the mosquito, but it doesn't bind the parasite. So it's, again, an example of a naturally occurring allelic variant or genetic variant that is present in the population as a whole, but one's way better from the point of view of humans than the other. And so we could, again, make scissors that cuts the parasite-friendly version and not the parasite-refractory version, and drive that into the population as well. So those are the kinds of things we're working on. And then to make it more acceptable, as you're talking about too, because there's some resistance, I think it's a debate that's going on, and my view is that the resistance is perhaps a bit stronger than it needs to be, but nonetheless, there's resistance to the concept of using full gene drives because of the obvious capacity of the system to spread itself through a population. And I understand why people are nervous about that, and one has to be very careful in terms of choosing the examples in which one would actually go forward with such a scheme. But given that, another option, particularly in the case of local areas, is to create a genetic system that only does the allelic modification, and that the genetic cassette, all built into one piece, is designed to disappear. And so we're doing that right now. We're testing that kind of approach right now. And the beauty of that is that you would add these insects to an environment. They would come in and mate with the other insects that are there. During the time that that genetic element is still present, it would do what you ask it to do, which is to shift the balance of the allelic variants, of the genetic variants, to those that you favor: insecticide sensitivity, parasite refractoriness. But then the genetic element would disappear because it's programmed to. You put it in a place where it's not good for it to be in the genome, and so you have a strong genetic selection for it disappearing. It disappears. Now it can't do its thing anymore, but you've made some bump up in the frequency of the genetic version that you want. You want more of what you had hoped to gain? Well, then you just add some more insects. You do it again. And so it's like treating with an insecticide, except it has no ecological damage at all. And the end result is the softest result you could possibly ask from a genetic transformation point of view, which is that you just transform one population to another, that one is a natural occurring population, and so is the second. It's just the second is the one that you prefer, an insecticide sensitive version or a parasite refractory version. And that is what we're most excited about, because we hope that that will get the backing, broader backing of the public, and that will also perhaps provide an entree for the more bold versions where you use a full gene drive to modify a population. Because when you think about doing things on the scale of a continent, that is what you would probably need.
That would be one more question I had, which was, to scale it up on a large enough scale, as Africa is a huge continent as well, for example, but also other places, Southeast Asia, etc. Is it feasible to do, in your view, long term, and how many years are we out?
It is, but I think in those situations, you really probably would have to use a full gene drive, because, as my colleague at UC Irvine, who has been a pioneer in molecular design of these insects, Tony James at UCI, has often pointed out, is that if you have a genetic system like the ones we're talking about, you can consolidate your gains. That is, you take the insects, you introduce them into a certain area, and because it's a stable and spreading genetic element, if you get it to work in a given area, it spreads throughout a given area, it stays put. So now if another insect comes in off of a ship or something like that, that's got the problem version, it's a malarial transmitting mosquito or something, then the existing mosquitoes that are present in the area, they'll act on that, copy their genetic information into it, and render it now a malaria refractory mosquito. And so you don't have to keep going back to the places that you treated. And if you make kind of the analogy with insecticides, that's the big problem also with insecticides, is that you keep having to use them. So even if you're successful and you totally wipe out the bad mosquitoes, in fact, that was done in the Western Hemisphere with insecticides back in the 50s. If you do that, you can wipe out all the insects you want, but eventually a ship comes back over with another batch of them, and then you have to do it all over again. And so the beauty of a gene drive is that you can, again, like Tony says, you can consolidate your gains, move on to another location, and keep then expanding the territory over which you have gained that control over the disease.
Ethan, really interesting listening to you, and great work. Thank you very much for sharing all this, and all the best for the further work. Thank you.
My pleasure. Thanks.